My dear students, welcome back to our channel. Students, in this video I am explaining Telangana Intermediate Second Year. Subject Botany. Part B. Short questions, 4 marks. Let's start quick revision. First important question is 1. Give a brief account of Bt cotton. Bt cotton is a type of cotton plant that has been genetically modified to fight pests, especially the cotton bollworm. Scientists insert a gene from the bacterium Bacillus thuringiensis, hence Bt, into the cotton. This gene helps the plant produce a protein that is toxic to certain insects but safe for humans and animals. For example, in a field with regular cotton, farmers might have to spray pesticides to protect the crop from pests. With Bt cotton, the plant naturally protects itself, reducing the need for these chemicals. This can save time, money, and help the environment. Bt cotton has been widely used in many countries, making it easier for farmers to grow cotton and improve yields. However, some people worry about its impact on biodiversity and the long-term effects of genetically modified crops. Next important question is 2. Write a note on agricultural horticultural applications of auxins. Auxins are plant hormones that play a key role in growth and development. In agriculture and horticulture, they have many useful applications. 1. Promoting root growth. Auxins are used in rooting powders to help plant cuttings grow roots. For example, if you cut a stem from a plant and dip it in auxin powder, the cutting will develop roots and become a new plant. 2. Fruit development. Auxins help fruit grow and mature. In some cases, auxins are sprayed on plants to encourage fruit set, meaning more fruit can develop. 3. Preventing premature falling. In some crops like apples, auxins are used to stop fruits from falling off the plant too early, allowing them to grow and ripen properly. 4. Controlling plant growth. Auxins can also be used to control unwanted plant growth, like preventing weeds from spreading. Overall, auxins help improve crop yield, make plants healthier, and aid in plant propagation. Next important question is 3. Explain different types of cofactors. Cofactors are non-protein molecules that help enzymes carry out their functions. There are 2 main types. 1. Inorganic cofactors. These are usually metal ions like iron, Fe, zinc, Zn, or magnesium, Mg. They help enzymes work by stabilizing the enzyme structure or assisting in chemical reactions. For example, iron is a cofactor for the enzyme that helps in oxygen transport in red blood cells, hemoglobin. 2. Organic cofactors. Coenzymes. These are vitamins or compounds that assist enzymes. A common example is NAD+, nicotinamide adenine dinucleotide, which helps enzymes in energy production in cells. Another example is vitamin B12, which acts as a coenzyme in processes like DNA synthesis. Both types of cofactors are essential for many biochemical reactions in the body, helping enzymes function correctly and allowing important processes like metabolism and digestion to occur efficiently. Next important question is 4. Explain the incomplete dominance with example, V.V.imp. Incomplete dominance is a type of genetic inheritance where neither allele is completely dominant over the other. As a result, the offspring show a mix or blend of both traits. For example, consider flower color in snapdragons. If a red-flowered plant, RR, is crossed with a white-flowered plant, WW, the offspring, RW, will have pink flowers. This happens because the red and white alleles blend together, and neither one fully dominates the other. So, in incomplete dominance, the traits don't disappear but combine to create a new, intermediate trait. It's different from regular dominance, where the dominant allele completely hides the effect of the recessive allele, like in pea plant color. Next important question is 5. Mention the advantages of selecting pea plant for experiment by Mendel. Mendel chose the pea plant for his experiments because of several key advantages. 1. Clear traits. Pea plants have distinct, easily observable traits like flower color, purple or white, seed shape, round or wrinkled, and pod color, green or yellow. This made it simple to track inheritance. 2. Self-pollination. Pea plants can self-pollinate, meaning they can reproduce on their own. This helped Mendel control the parent plants and predict the traits of the offspring. 3. Quick growth. Pea plants grow quickly, allowing Mendel to observe several generations in a short time. This made his experiments more efficient. 4. Easy to crossbreed. Mendel could easily cross-pollinate pea plants by transferring pollen between different plants, which helped him study how traits are passed on from one generation to the next. These advantages allowed Mendel to develop his laws of inheritance, which laid the foundation for modern genetics. Next important question is 6. Write the important features of genetic code. The genetic code is a set of rules that cells use to translate information from DNA into proteins. Here are its important features. 1. Universal. The genetic code is the same in almost all living organisms. For example, both humans and bacteria use the same code to make proteins. 2. Triplet code. It is made up of sets of 3 nucleotides, DNA building blocks, called codons. Each codon codes for one amino acid, which is a building block of proteins. For example, the codon AUG codes for the amino acid methionine. 3. Redundant. Some amino acids are coded by more than one codon. For example, both GCU and GCC code for the amino acid alanine. 4. Non-overlapping. Each nucleotide is part of only one codon. It does not overlap with the next one. 5. Start and stop signals. Specific codons signal the beginning, AUG, and end of protein synthesis. These features ensure that proteins are made accurately and consistently in cells. Next important question is 7. Explain the steps involved in the formation of root nodule. The formation of root nodules occurs when certain plants, like legumes, example, peas and beans, form a partnership with nitrogen-fixing bacteria. Here are the steps involved. 1. Bacterial attraction. The plant releases chemicals called flavonoids, which attract nitrogen-fixing bacteria, like rhizobium, to the roots. 2. Bacterial infection. The bacteria enter the root hairs through a small opening and move inside the root cells. 3. Nodule formation. The plant's root cells respond by dividing and forming a small swelling, known as a nodule. This is where the bacteria will live and fix nitrogen. 4. Nitrogen fixation. Inside the nodules, the bacteria convert nitrogen from the air into a form, ammonia, that the plant can use for growth. 5. Symbiotic relationship. In return, the plant provides sugars and other nutrients to the bacteria, forming a mutualistic relationship. This process helps plants grow in soils with low nitrogen, benefiting both the plant and the bacteria. Next important question is 8. Draw a neat labeled diagram of chloroplast. Next important question is 9. What is ICTV? How are viruses named? ICTV stands for the International Committee on Taxonomy of Viruses. It is an organization that classifies, and names viruses based on their structure, genetic makeup, and how they infect hosts. The ICTV helps ensure a standardized naming system for viruses around the world. Viruses are named using several rules. 1. Family and genus. The virus is first placed in a family, which is a group of related viruses. For example, the family Coronaviridae includes viruses like the one causing COVID-19, SARS-CoV-2. 2. Species. Within each genus, viruses are given a specific species name based on the disease they cause or the organism they infect. For example, HIV-1 is the species causing AIDS. 3. Descriptive terms. Virus names can describe their shape, host, or location. For example, influenza A virus causes flu, or tobacco mosaic virus infects tobacco plants. This systematic naming helps scientists understand and study viruses. Next important question is 10. Define RQ. Write a short note on RQ. RQ stands for respiratory quotient. It is a number that helps to understand the type of fuel, carbohydrates, fats, or proteins the body is using for energy during respiration. RQ is calculated by dividing the amount of carbon dioxide, CO2, produced by the amount of oxygen, O2, consumed. The formula is RQ is equal to CO2 produced, O2 consumed. Example: when the body uses carbohydrates for energy, the RQ is about 1.0 because the amount of CO2 produced is equal to the amount of O2 consumed. When the body uses fats, the RQ is about 0.7 since fats require less oxygen to produce the same amount of energy. Proteins have an RQ around 0.8. RQ helps scientists and doctors understand metabolism and energy use in the body, and it can also indicate the type of fuel being used during exercise or rest. Next important question is 11. Write briefly on nucleosomes. Nucleosomes are the basic units of chromatin, which is the material that makes up chromosomes in the nucleus of a cell. They help package DNA so that it fits inside the cell's nucleus. A nucleosome consists of a segment of DNA wrapped around a core of proteins called histones. These histones act like spools, and the DNA wraps around them to form a structure that looks like beads on a string. For example, imagine the DNA as a long thread that needs to be tightly packed. The histones are like spools, and the thread is wound around them to create a compact structure, making it easier for the cell to store and organize the DNA. Nucleosomes play a key role in controlling gene expression because they can help regulate which genes are accessible for being read by the cell, affecting how proteins are made. This process is important for cell function and growth. Next important question is 12. Explain the conjugation in bacteria. Conjugation in bacteria is a process where one bacterium transfers genetic material to another through direct contact. This is a way for bacteria to exchange DNA, often leading to genetic diversity. Here's how it works. 1. Pilus formation. One bacterium, called the donor, forms a structure called a pilus that connects to another bacterium, the recipient. 2. DNA transfer. The donor bacterium transfers a small piece of DNA, usually a plasmid, a small circular DNA, to the recipient through the pilus. 3. Genetic change. The recipient bacterium receives the new genetic material and can use it, which may give it new traits, like antibiotic resistance. For example, E. coli bacteria can exchange plasmids carrying genes for antibiotic resistance, making the recipient resistant to antibiotics. Conjugation is an important way for bacteria to share useful traits quickly, helping them adapt to changing environments. Next important question is 13. Write briefly about enzyme inhibitors. Enzyme inhibitors are substances that slow down or stop the action of enzymes. Enzymes are proteins that speed up chemical reactions in the body. Inhibitors can prevent enzymes from working properly, which can affect many biological processes. There are two main types of enzyme inhibitors. 1. Competitive inhibitors. These molecules compete with the substrate, the molecule the enzyme acts on, for the enzyme's active site. For example, methotrexate is a competitive inhibitor that blocks the enzyme involved in cell growth, used in cancer treatment. 2. Non-competitive inhibitors. These bind to a different part of the enzyme, changing its shape and making it less effective. For example, cyanide inhibits enzymes involved in energy production in cells, which can be harmful. Enzyme inhibitors can be natural or synthetic, and they are used in medicine to control enzyme activity, like in antibiotics or cancer drugs, to treat diseases. Next important question is 14. Describe in brief photorespiration. Photorespiration is a process that occurs in plants when they take in oxygen and release carbon dioxide, unlike regular photosynthesis, which takes in carbon dioxide and releases oxygen. It happens when the enzyme rubisco, which usually helps plants take in carbon dioxide during photosynthesis, mistakenly binds with oxygen instead. This creates a wasteful reaction, as it reduces the plant's ability to produce sugar, which is essential for growth. For example, in hot and dry conditions, plants close their stomata, tiny pores, to prevent water loss, but this also reduces the amount of carbon dioxide entering the plant. As a result, rubisco ends up using oxygen more, leading to photorespiration. Photorespiration is inefficient because it wastes energy and reduces the plant's ability to make food. C3 plants, like wheat and rice, are more affected by photorespiration, while plants like corn and sugarcane, known as C4 plants, have developed ways to minimize this process. Next important question is 15. Explain the structure of T11 bacteriophages. T11 bacteriophages are viruses that infect bacteria, particularly E. coli. They have a simple structure designed to attach to and inject their genetic material into the host bacteria. Here's a breakdown of their structure. 1. Head. Capsid. The head is a protein coat that contains the viral DNA. It looks like a hexagonal or icosahedral shape. The DNA inside is the genetic material that will be transferred into the bacterial cell. 2. Sheath. Attached to the head is a long, contractile sheath. This part contracts when the virus infects a bacterium, helping inject the viral DNA into the host. 3. Tail fibers. At the bottom of the sheath are tail fibers, which are used to attach the bacteriophage to specific receptors on the surface of the bacterial cell. 4. Base plate. This structure helps anchor the bacteriophage to the bacterial surface and aids in DNA injection. T11 bacteriophages, like T4, are important for studying how viruses infect cells and replicate. Next important question is 16. What are the differences between DNA and RNA? DNA and RNA are both types of genetic material, but they have some key differences. 1. Structure. DNA, deoxyribonucleic acid, is double-stranded, like a twisted ladder, double helix. RNA, ribonucleic acid, is single-stranded. 2. Sugar. DNA contains deoxyribose sugar. RNA contains ribose sugar. The difference is that ribose has one extra oxygen atom compared to deoxyribose. 3. Bases. DNA uses the bases adenine, A, thymine, T, cytosine, C, and guanine, G. RNA uses adenine, A, uracil, U, cytosine, C, and guanine, G. Instead of thymine, RNA has uracil. 4. Function. DNA stores genetic information and is found in the nucleus in eukaryotes. RNA helps carry out the instructions from DNA, mainly in protein synthesis. RNA is found in both the nucleus and cytoplasm. For example, DNA holds the blueprint for making proteins, while RNA helps build those proteins in cells. I hope you understand easily each question. All the best, students, for your upcoming exam. For other subject quick revision. Important questions. Previous question papers. And syllabus. Links are available in description. Check out once. Thank you for watching. Like and share this video with your friends.